All right, welcome back to the deep dive. Today, we're going deep on something pretty crucial, I think. It's bacterial resistance to antibiotics.
Yeah, it's a really growing threat, and to even begin to address it, we have to understand how how the bacteria are fighting back against us.
Yeah, absolutely. It is a constant arms race,
Yeah,
right, between the drugs that we develop and the ways that the bacteria evolve to resist them. So our mission in this deep dive is to give everybody a clear view or a clear picture of the core strategies that these bacteria are actually using to evade these essential medicines.
that's right. And to do that, we're going to be taking a look at some of the latest scientific research that outlines the mechanisms of resistance.
Okay, so let's start with, I guess, the most direct approach, right? What's called enzyme inactivation. So this is basically the bacteria deploying, a chemical weapon of some sort.
Right, it's a chemical defense to disarm the antibiotic.
Yeah.
Right? So what happens is that the bacteria produce enzymes, right? And these enzymes can directly attack and disable the antibiotic molecule.
Interesting.
And so a really good example of this is something called beta-lactamases.
Okay.
And these beta-lactamases break down a really key structure that's in a lot of antibiotics called the beta-lactam ring.
Right.
And this beta-lactam ring is found in a lot of very common antibiotics like penicillins, cephalosporins, things you've probably heard of. And what's really significant is that there's a huge variety of these beta-lactamases,
Mhm.
and different beta-lactamases can target different classes of drugs even. And even worse is that the genes that code for these beta-lactamases are often found on mobile genetic elements
Oh, interesting.
called plasmids.
Okay.
And these plasmids allow the bacteria to share these resistance genes really rapidly.
So it's like they can share the blueprints for uh, how to, disable these different antibiotics. That's really remarkable. What about other types of antibiotics? So, like the aminoglycosides, for example.
Yeah, so for those types of drugs, like the aminoglycosides, bacteria use something called aminoglycoside modifying enzymes,
Okay.
AMEs.
All right. So AMEs.
And what they do is they'll add chemical tags to
Yeah.
the aminoglycoside molecule, and this basically prevents it from binding to the ribosome, which is
Mhm.
its target in bacteria. And again, the genes for these modifying enzymes are often located on mobile elements, so they can be shared easily between different bacteria.
So this ability to share these resistance genes is a huge factor, it sounds like.
Oh, yeah, absolutely. Yeah.
What other sorts of enzymatic defenses do these bacteria have? We've talked about beta-lactams and aminoglycosides, but
Right. So there's also something called chloramphenicol acetyltransferases.
catchy name.
Yeah, we call them CKATEs for short. And they inactivate chloramphenicol, which is another antibiotic, through a similar modification strategy that prevents it from binding to the ribosome. And then there are also esterases, which some bacteria use to inactivate macrolides, but this is a bit less common.
Okay. Okay. So that's really interesting. So they're directly, basically neutralizing these drugs.
Mm,
Now, there's another way that they can also fight back,
right.
right? And that's by actually changing their own internal components so that the drugs can't bind effectively.
Right.
So this is what's known as target site modification.
Exactly. So instead of attacking the antibiotic directly, what the bacteria can do is they can actually alter the specific molecules within their cells that the antibiotic is designed to interact with.
Interesting.
So as an example, penicillin binding proteins, or PBPs,
Right.
mutations in the genes that code for these PBPs can actually change the shape of the protein itself.
Right.
And by changing the shape of the protein, it makes the beta-lactam antibiotics less effective at binding. And so a good example of this is MRSA, which is methicillin-resistant Staphylococcus aureus.
Yeah.
And it's acquired this new PBP called PBP2A that's resistant to many beta-lactams.
Wow.
And then on top of that, the ribosome itself can be modified too.
Oh, wow.
So what can happen is that the ribosomal RNA, specifically the 23S ribosomal RNA, it can be methylated by enzymes called methyltransferases, and that methylation can prevent macrolides, lincosamides, and streptogramin B from binding to the ribosome.
Okay.
And again, these genes are often found on those plasmids.
Wow. It's incredible how they can share these genes.
Yeah, it's really pretty remarkable.
So they're changing the actual machinery that the antibiotics are trying to target.
Huh.
So what about, other essential processes in the bacteria, like, Something like DNA replication or RNA transcription.
Right. So bacteria can also modify enzymes like DNA gyrase and topoisomerase 4, which are the targets for fluoroquinolone antibiotics.
Yeah.
And what happens is they get mutations in the genes that code for these proteins, and it reduces the drug's effectiveness.
Okay.
And then you can also get mutations in RNA polymerase, which can lead to resistance to rifampicin, which is another antibiotic.
Okay.
And then, really importantly, in vancomycin resistance, what happens is that the bacteria actually modify their cell wall precursors, and so they change the target. So the vancomycin can no longer bind and do its job.
It's really a constant, game of adaptation, isn't it? It's really incredible. So we've talked about, a couple different things. We've talked about enzyme inactivation. We've talked about target site modification. Now, another thing that bacteria can do is they can actually control how much antibiotic even gets inside the cell in the first place, and they can even pump it back out.
Exactly.
So this is what's referred to as reduced permeability and increased efflux.
Right. So gram-negative bacteria, they have this outer membrane with porin channels that allow for the entry of some antibiotics, but changes in these channels can restrict the entry of drugs like beta-lactams and fluoroquinolones. And then there's also these efflux pumps, which are membrane proteins that basically actively pump the antibiotics back out of the cell.
So it's not even just blocking the entry, but also just kicking them back out.
Yeah, that's right. They're really good at it. And many of these pumps can actually expel multiple types of antibiotics, which is what contributes to this phenomenon of multi-drug resistance.
Right.
And some of the key players in this are families like the RND family and the MFS family. And what's even more incredible is that their activity of these efflux pumps can increase in response to antibiotic exposure.
Wow. So they're sensing that it's there, and then they're increasing the activity of these pumps.
Exactly.
That's amazing. All right. So we talked about enzyme inactivation, target site modification, reduced permeability and efflux. And then I guess finally, sometimes the bacteria can sort of just find a completely different way to function when a pathway is blocked, right? So they can just kind of bypass it altogether.
Right. So this is what we call target or metabolic bypass.
Okay.
So bacteria can acquire genes for resistant versions of essential enzymes.
Okay.
And so an example of this is enzymes that are involved in folate synthesis.
Uh-huh.
And so this is how they become resistant to drugs like trimethoprim and sulfonamides.
Interesting.
And then less commonly, they might also overproduce the target enzyme.
Okay.
Or, and this is very rare, but they might even develop, entirely new metabolic pathways.
So they can just sort of, evolve a new way of doing things.
Hmm.
That's incredible. It's kind of amazing how many different ways that these bacteria have evolved to become resistant. And as we've been talking about, I mean, a single bacterium can have, multiple of these mechanisms.
Exactly. So they're often not relying on just one strategy. They're using many at the same time.
Yeah.
That's actually what makes treating some of these infections so difficult.
Yeah, absolutely. So just to recap, we've covered enzyme inactivation, target modification, reduced permeability and efflux, and target/metabolic bypass. It really just highlights the complex battle that we face when it comes to treating bacterial infections.
Yeah, it's a constant arms race, and understanding these strategies is really crucial if we're going to stay ahead.
Absolutely. Well, this has been a really fascinating, I think, look into the sort of intricate ways that these bacteria can evade antibiotics. It really makes you consider how, now that we know some of these tactics that they're using, how can we develop innovative new approaches, whether they're new types of antibiotics or even alternative therapies? How can we outsmart these microbes? It's a huge question for the future of medicine, and I really appreciate you being here to explore that with me.
Oh, it's my pleasure.
Thanks for joining us on The Deep Dive.